Bismillahirrahmanirrahim. Assalamu alaikum. How are you all? I welcome you in Pakistan International School Types virtual learning system for the session 2020, and this is biology class for grade first year. Dear students, today we will see lecture number 24. In that we will continue our chapter number 3, enzymes. And in chapter number 3, enzymes, as you know that we have studied a detailed account of enzymes, in which we have studied about its structure, its properties, and some of the factors which affect the activity of enzymes, and we have seen enzyme inhibition. Today we will see feedback inhibition and classification of enzymes. So let's get started. Dear students, if we see feedback inhibition, so you have to recall the previous mechanisms of enzymes inhibition, in which we have studied about competitive and non-competitive inhibition. And in that we have seen that non-competitive inhibition is irreversible or reversible as well. And the reversible non-competitive inhibition is called feedback inhibition. So how this feedback inhibition works? Because this feedback inhibition that goes all around in our body to regulate enzymes function. So enzymes are regulated by their own products when they are more than its required quantity. So let's take a look on an example that we are having here. Like this is an enzyme E1. So we can take example of this enzyme, like this is enzyme, and enzyme E1, and it will act like that is what we can say aspartate. It acts on this amino acid aspartate, and this aspartate enzyme will change into threonine amino acid, but it just carries on lots of here like lots of intermediate reactions. So it does not go directly from aspartate to threonine. So it just go from aspartate to another that is called intermediate product A. Then this intermediate product A that is converted into B, but it's going on the hands of several other enzymes. So enzyme E1 will change this product into another product that is we can say here at this place aspartate. By the action of enzyme 1, it is changed as aspartokinase. Then with the help of this product, intermediate product, with the handing over to another enzyme, and this will be changed into another product, aspartyl B semi-aldehyde dehydrogenase, and then it will go to another pathway and handed over to another enzyme 3, and we are getting intermediate product C, that is we can say homoserine kinase enzyme acts over there. Then it is changed into another intermediate product, and enzyme 5, that is called threonine synthase act over it. So at number E1 is aspartokinase enzyme, E2 is aspartyl B semi-aldehyde dehydrogenase, like hydrogen is removed from here. Then at number 3 enzyme we are having here hemoserine and dehydrogenase, then we are having at number E homoserine kinase, and then number E6, A5, sorry, we are having threonine synthase, and in the last we are having our final product threonine. Enzyme just catalyze the reaction up to the requirement when the lots of threonines are produced, so they can proceed the reaction, uh, and when they are more in number, so what happened, these products will move to the allosteric site of the enzyme to bind with the enzyme, and when allosteric enzyme, this product will bind, so what happened, mm, this in this substrate which has to be changed into threonine, this will not be able to bind with active site of enzyme E1, that is aspartokinase. So it means when allosteric bind site is busy, so active site will not be able to bind any type of substrate. So the production of aspartate to threonine is going to be stopped, because there is enough amount of threonine in the body is present. So it is called feedback inhibition, like from the back they are just gonna inhibit the process. This is also having very helpful and useful role to regulate the enzyme activity. And when threonine going to be reduce in the body, so what happened, this allosteric site will be free and threonine will be um, released from here, from allosteric side, so active side of this will be again bind with the aspartate, so the reaction will proceed again. So this is feedback inhibition or negative feedback, we can say, and that can be happened with lots of metabolic processes which are going on in the body. So you can see here feedback inhibition in this diagram, that how the inhibition, enzyme inhibition is going to be happen. Like this is an a substrate which is bind with this active site of enzyme, so it will proceed further reaction. Here can be the intermediate product, then this intermediate product is going to be handed over to another enzyme, and then that produce another intermediate product, and then this bind to another enzyme and make the final product. So if they are in more quantities, so these products again bind with enzyme number 1 and with the allosteric side, so that shuts down the pathway of production of this product here. So this regulates the activity of enzyme actions. So dear students, let's talk about our next topic of this chapter. Actually, this is the last topic in which we will study the classification of enzymes. Enzymes are classified either on the reaction types on which they catalyze the reaction, and well as they are having the classification on the substrate basis. So let's talk about one by one. So you can see here classification of enzymes which are based on the reaction they catalyze. So there are six classes of enzymes which are based on their chemical reactions, like oxidoreductases, which do oxidation and reduction in our body. Transferases, they do transfer grouping of atoms, excluding H group. They do not transfer hydrogen group. So another any functional group, they transfer from one molecule to another. Hydrolases, by addition of water, hydrolysis is done, and we can break the molecules, larger polymers. Then we are having liases. Liases means they add atoms or remove atoms, or they form a double bond between them. We'll see one by one in detail. Isomerases, they rearrange the atoms and they form the same molecular formula. They will can change one molecule to the other form. Iso means same, so they form the same molecules. So ligases, they join or they combine the molecules and make the polymers, and they use ATP in that. So if we see another classification that is based on the substrate, uh, so we can have, which is like there are lots of enzymes which based on a reaction based, synthesis based, chemical nature, substrate based. So we have seen reaction based, now we'll see substrate based as well. So enzymes, when they are substrate based, so there is A's at the end, so we just remember that they are enzymes. So there are carbohydrates, uh, like carbohydrates. They are like reacted by or catalyzed by carbohydrates. Then we are having proteases, yeah, proteinases, which act on proteins, lipases, which act on lipids, and nucleic acids, which acts on um, like nucleases, which act on nucleic acids. Then maltose, um, then we can have like maltases, lactose on lactose, lactase hydrolyze the reaction, and sucrase hydrolyze sucroses. So let's talk about them one by one. Okay. So when we just talk about on the base of reaction, so that is the names are given by International Union of Biochemistry, like IUB, and in 1961. So they are categorized in 6 categories. So they are oxidoreductase, the first one. We know that in our body, the metabolic activities, they can be oxidation, there can be reduction, means they can lose the electrons or they can gain the electrons, they can lose the protons, or they can lose the proton or adding of proton in that. So that is oxido from oxidation, reductor from reduction. So these enzymes do oxidation reduction reactions. For example, a cytochrome oxidase oxidizes a cytochrome, which is the intermediate compound in the respiration. Then we are having transferases. So transferases means, name is telling you that the enzymes catalyze the reaction in which the units, functional units, are going to be transferred from one molecule to another molecule. So for example, this example is hexokinase, and that does its function in respiration. So it transfers phosphate group from ATP to glucose, so it makes glucose 6-phosphate. It means from taking phosphate from ATP, what it does, it gives phosphorus to glucose. So we will see the next one, that is hydrolysis. Hydrolysis, definitely they do hydrolysis. And with the addition of water, means with the hydrolysis process, what happens, the larger molecules, they are complex molecules, they break down into smaller one by adding hydrogen H or H group of water. So lots of proteolytic enzymes which work on proteins, so they can break down the proteins into smaller peptones or peptides. Peptones are actually the soluble proteins, and that is formed in the early digestion of proteins. Means they are not smaller amino acids or dipeptides, but they are larger broken molecules of proteins, which are then changed into peptides and then into amino acids. So we can see that we are having lots of enzymes: pepsin, renin, trypsin. They break up or perform the function of proteins by the by help or by the use of hydrolysis enzymes in the process of hydrolysis. Okay, if we see lysases, so these enzymes catalyze the breakdown of specific covalent bonds, and they remove some of the bonds or atoms or molecules or even the groups from those complex molecules, but without hydrolysis. Like here in this hydrolysis, addition of water is done and then the larger molecules are broken down, but lyases without hydrolysis process, groups are broken down, means covalent bonds are broken down, and those functions are performed by lyases. And for example, histidine decarboxylase, it breaks the covalent bonds in histidine and remove carbon dioxide and histamine. Histamine actually is immune, it gives immune response, and that is neurotransmitter. So what happened? Decarboxylase means it removes carbon dioxide from histidine, then it becomes histamine and carbon dioxide. In isomerases, we know that enzymes, what they do the intramolecular rearrangement. So molecules, for example, these are the molecules which are arranged by this. They are having same formula, molecular formula, sorry, chemical formula, but they can have the different structure. So this this molecule can be broken down by this, we can say, by this covalent bond, and it can be arranged here. So it means that they can have the structure like this. So the molecules are rearranged in different forms by rearranging the molecules. That is done by isomerases, and we can have the example of phosphohexokinase isomerase. The name is telling you that it just isomerase the phosphate group, because before we have studied that it will form hexokinase form glucose 6 phosphate by removing one phosphate from ATP and adding it into the glucose at a carbon number 6. So this glucose 6 phosphate is going to be changed into fructose 6 phosphate. So the molecular number is changed, but rearrangement is different. Ligase word comes from glue, like they join or synthesize the molecules. They join smaller monomers or dimers and they make polymers, long chains. So they act upon mostly by using the energy of ATP. Hydrolysis of energy, hydrolysis of ATP is done, and ATP is changed into ADP plus P. So when the bond is broken down, so what happened? The energy utilized for that this process, and that process is done by polymerases, which are the enzymes which perform the function for polymerization or making the big polymers of DNA and RNA. So let's take a brief look on the reaction procedure, like how oxidoreductases perform. If one is reducing and other is oxidizing, so they can change like this: from one electrons are removed and other is taking. So one product will be oxidized, the other will be reduced. Transferases, we transfer between two products one molecule from here to the other molecule, so it can be A plus C bond B. Hydrolysis is done by hydrolases, so they perform the addition of water in between. So A and H bond will be formed and B and OH will be formed. It means these two molecules, the covalent bond which is in between them, it is broken down. Lyases, it breaks down without hydrolysis. So what they do without hydrolysis with the using of energy, so they form A, B compounds. Isomerases, they do the rearrangement. You can see here A, B, and C. So they form A, C, and B, like C transfer to between A and B. Ligases or synthetases, we can also give them synthetases. So they join monomers or dimers and form polymer chains uh, of long molecules of especially DNA and RNA and proteins, and they use ATP, and ATP is broken down into ADP and PI. Let's talk about the classification based upon substrate. So the name is telling you actually that this enzyme just functions on proteins and which type of substrate it is. Like the first of all, we see proteases. They can also be said as peptidases or proteinases, like they functions on the proteins. So we are having pepsin, trypsin, both functions, both digest large polypeptide chains into smaller polypeptide chains and peptones. We have told about peptones and polypeptides. Then aminopeptidases and carboxypeptidases both digest peptones and dipeptides, and then eripsin digest dipeptides into amino acids. So this is a cascade or long chain reactions. First of all, we can see here we are having large or long polymers of peptidases on that pepsin attacks. This is number one. Okay, pepsin and trypsin attacks. This is number two, attacks on these large polypeptides which just come into our stomach. Then what happened? They are broken down into polypeptides and peptones. These are intermediate products. Now these intermediate products are handed over to aminopeptidases. This is the third enzyme. Aminopeptidases, what it does, it further, and carbopeptidase, oxypeptidases, it further breaks down these peptones and peptides into dipeptides, because these are still long chains, into dipeptides. So this is another intermediate product. Now what happened? Dipeptides are handed over to eripsin. So eripsin is the enzyme of which digest or break down these dipeptides into amino acids. So we know that amino acid is a unit of proteins. So next we will see lipases. Lipases, your name is telling you, acts on lipase, and it is happening in pancreatic juice. We secrete pancreatic lipases to digest lipids into fatty acids and glycerols. We know that glycerols and fatty acids units of lipase, lipids. Then we are having carbohydrates. Carbohydrates, because carbohydrates are present in many forms. So according to that substrates, we are having different enzymes like amylases that digest starch and glycogen into maltose. To maltose is actually disaccharide, and it is um, the molecule of glucose plus glucose. So that is disaccharide. Then the disaccharides are broken down into further monosaccharides. Then we are having cellulases. Cellulase is also digest with the help of with the help of cellulase. So into cellubiose. So that is also disaccharide. Maltases, they digest this maltose into glucose. So it means that is broken down into glucose, that is monomers. Sucrase, that's broken down sucrose into sucrose is disaccharide. So it breaks then into glucose and fructose. These are monosaccharides. Lactases, they break down lactose, that is also disaccharide, and it's broken down into galactose and glucose. Then we will see nucleases, some of the nucleases. So when we talk about nucleases, so nucleases are those enzymes which break down DNA or RNA. So RNases, as the name is telling you, digest RNA into ribonucleotides, like into the small monomers of ribonucleotides. As we know that nucleotides are the monomers or units of RNAs and DNAs. Then DNases are they digest these enzymes does inorganic phosphate, and this process in the muscles going on again and again. Dear students, I hope you understood this lesson also, and it is also very important because related to our body, as well as we will finish with this topic our chapter number 3, enzymes. Inshallah, we will continue with the new chapter, new topics in the next lecture. Wish you all the best. Allah Hafiz.